So a pleasant afternoon to each and every one of you. My name is Serafin from the Philippines. So it's a pleasure to be talking in front of you. So it's literally, I'm standing upon giants of my fellow colleagues. So I'm here to present our paper on the thermal influences of the life cycle of the upside-down jellyfish, Cassiopea spp, in a tropical shallow embayment. As we all know, climate change is a recent topic in the recent decade, specifically on global warming. So predictions indicate that increasing sea surface temperatures will have various effects on different biological and ecological systems, notably in our case, gelatinous zooplanktons. Contrary to common assumptions, the association between global warming and the increased occurrence and abundance of jellyfish blooms have not been fully understood yet. Current extensive research and reviews have revealed that responses would vary among species and populations, and even within the same species across varying geographic regions. While a significant body has been focused on the temperate species, there is noticeable scarcity with knowledge regarding to population dynamics in the tropical region. So organisms in this tropical region are said to be living within or close to their thermal threshold and having adapted to a narrower and stable temperature range. So this study focused on a population of upside-down jellyfish situated in a very shallow embayment in Maribago, Cebu, Philippines. Though this population is morphologically the same, it actually comprises of two species, Cassiopea mayeri and Cassiopea colinensis. The habitat is characterized by a rocky sandy seagrass bed subjected to a very low tidal variation of 0.15 to 0.65 meters. The in-situ temperature measurements taken last May 2016 has revealed a range of 28.7 degrees to as high as 34.4 degrees, with an average of 31.9 degrees. So in this area, there is notably medusas are found all year round, with females with brooded embryos in their central oral arms. Interestingly, despite their year-round appearance, there is substantial variation in size, frequency, and abundance each month. Notably, there is higher abundance of smaller size medusa, less than 6 centimeters, which are occurring during and after the northeastern monsoon, which brings colder winds from Siberia. So this study focuses on investigating the responses of this tropical upside-down jellyfish to a very narrow range of warm temperature. Specifically, we wanted to check or assess larval settlement rate from the sexually produced planulae larvae. At the same time, examine sexually reproductive responses of the benthic stage, specifically the strobilation and budding. We initially hypothesized that these responses will not vary across this very narrow range of temperature. As for the methods, we started off with collecting female medusas with visible pinkish to orange embryos within their oral arms, and then we isolated them individually and left them in the dark for 36 hours, after which actively swimming planulae larvae was then collected and pooled for succeeding analysis for Objective 1. And for Objective 2, a polyp culture was started to test for the strobilating and budding experiment. So our chosen temperature range from 27 to 35 degrees was based on the in-situ measurements taken, with 27 being the least warm and 35 being the warmest. So for the first objective, settlement, actively swimming planulae larvae were individually collected and placed individually in plastic containers. Before the start of the experiment, a preconditioned floating polystyrene film was then added. The entire experiment was set to 5 days with the following observation time points. So we also had biological and technical replicates. The percent settlement count was calculated at each time point, and an individual was considered settle when successful formation of the pedal disc and the two tentacular bud. As for the second objective, for the settlement and budding, mature polyps were isolated from the culture and then transferred to individual containers and was left to reattach for about 1 week in the dark. So after which, any unattached polyps or dead polyps were then removed from the containers. The entire experiment was set for 30 days with daily observations, and the setup was fed with 3 times a week with fresh hatch Artemia nauplii ad libitum in order to saturate feeding with a 12-12 light-dark cycle. So we have also both biological and technical replicates. since some of the polyps had died, so there was an uneven number of polyps per container per treatment. So attributes that were measured was the percent strobulating polyps and budding polyps per day. So all statistical analysis was conducted in RStudio or R and passed for. So for the results for this first one, we can see that as there was a significant impact between time, temperature for larval settlement. As we can see that as time progressed and as temperature increased, settlement also increased. Wherein if you look at it, 35 degrees had the most increased number of settled numbers, followed by 33 degrees. While 27 to 31 degrees, there was no significant difference among this range. Notably, within the 5 days period, there was no 100% settlement observed in any of the treatments. At the same time, no mortality was observed. Looking at the 35 degrees planulae that were still alive, they had a very shrunken appearance. At the same time, they were moving or swimming sluggishly at the bottom of the container. While temperatures less than that, they were still actively swimming. So this enhanced settlement could possibly be due to increased metabolic demand due to warmer temperatures. As we all know that Cnidarian planulae don't have an oral mouth, so they rely on their embryonic stored energy. So the warmer the water, the faster the metabolism, the more there is need for them to settle and metamorphose into polyps so that it can start feeding. So this is the results of this study is also comparable with other species in the temperate area for Cyanea lamarcki and Chrysauria hysocelia. At the same time, in the Mediterranean for Cotylosa tuberculata, wherein faster or enhanced settlement was brought about by warmer temperatures. At the same time, it's also comparable to Cassiopea samachana in the Florida Keys, wherein within the range of 15 to 33 degrees, there was no change in the settlement rate. So in this case, for this study, it's 27 to 31, there was no change. But there was a greater settlement rate for temperatures greater than 33. So for the budding and strobulation experiment, it was observed in all treatments except for 35 degrees. So monodiscus was observed for strobulation, and budding accounted for single chain bud to multiple chain bud found at 33 degrees only. So looking at the cumulative count for each of the temperature range, we can see that there is an inverse pattern, wherein at more warmer temperatures, more buds were produced, while at lower temperatures, ephyra production dominated. Notably, one reason for the increased number of bud released in 33 degrees was the formation of chain buds in the base of the calyx, wherein as much as 3 buds were formed at the base. So looking at the ANOVA for the strobulation experiment, we can see that there is a significant difference between 27, 28, and 29 to 33 degrees. So there's no difference between 27 to 31, and 33 degrees had a lower percentage of strobulation. So this could mean that within this range, or basically the entire year round in the Philippines, strobulation can occur. More specifically on the cooler months during the northeastern monsoon where waters are relatively less warmer. So this also agrees with the same studies in Cassiopea andromeda and Samachana, wherein within this temperature range, strobulation was more enhanced. So at the same time, so we can see that in 33 degrees, strobulation was less. So it could mean that during the summer months wherein it's way hotter in this shallow embayment, so strobulation might be arrested or limited during this time. For the budding results, we can see that there is statistically no difference among the 4 temperatures. But visually looking at the graph, we have more percentage of budding polyps that are found at 31 and 33 degrees. So this could possibly mean that within this range, since all polyps had budded, so budding can possibly occur all throughout the year within the shallow embayment, but most specifically during the warmer months. But there's still more study that can be done. But in contrast to that, for Cassiopea andromeda in the Red Sea, the researchers have found that budding was more prominent in cooler temperatures at 17 to 18 degrees compared to warmer temperatures of 20 to 30 degrees. Looking at other species like Aurelia aurita, wherein they are known to exhibit varying responses at different geographic range. So there was enhancement of budding at warmer temperatures at the Baltic and Mediterranean Sea, while the opposite was found in Taiwan, where in cooler waters, budding was enhanced. But really looking at it, they found that in the Baltic and Mediterranean Sea, it says more warmer temperatures, but it's technically the cooler temperatures of Taiwan. So they still move around the same mean of 20 to 21 degrees. Although in my study, mortality wasn't part of the objectives, so I can like to present this without a rough statistical exam. We can see that for the first 4 temperatures, 27 to 33 degrees, they more or less have the same survivability rate. But looking at the 35 degrees, mortality dropped to 100% about the 15th day. So in my observation, the cause of mortality was a form of an eversion wherein the polyp would flatten itself and then spill open its guts. And then, I don't know what happened. Eventually, it would lead to the polyp being unable to feed. At the same time, disintegration. So this was found all over all treatment. So in conclusion, I would say that there is enhanced settlement at more warmer temperatures, leading to increased benthic recruitment during the summer months from March to May in the country. At the same time, strobilation and budding can occur all year round within the range of 27 to 33 degrees. But each of this asexual trait might increase or decrease during specific months. At the same time, I'd like to point out that for the polyp stage, there is a survival threshold for 35 degrees and greater, and that strobilation might be limited at 33 degrees or more. So I'd like to say that this Cassiopea in this country, or in this part of the world, are insensitive to small thermal variations from 27 to 31, but temperatures greater than this, the polyp stage or even the larval stage can become sensitive to their behavior already. So this study is without its limitation. At the time of collection, no molecular analysis was done. So initially we thought it was only one species. So it could be a source of variability within the data. We also had a smaller sample size due to constraints in resources and in the facilities. And of course, I had a smaller observation time for settlement, 5 days compared to other studies which had like 50 days or 200 days longer. And at the same time, a limited observation time for the benthic asexual traits. At the same time, I also lack a in-depth examination of the mortality data, so I still have to consider that. At the same time, there's a lack of more robust ecological and population data on this species. So with that, I'd like to recommend more extensive baseline data with all Scyphozoan species within our region. At the same time, to explore the lower thermal limit for these tropical species, and more studies on population dynamics and how they are being driven within the region. At the same time, of course, with that, you have to study their reproductive biology as well. So I would like to thank the following people for the support of this research. At the same time, the University of the Philippines to support this travel to India and making me talk to each one of you. Thank you.